I will. Hello, can you see my screen?
Hello, yes, we can see it.
Sorry. Thank you, Samira, for giving me the opportunity to present to you what we are doing here in Senegal for the diagnostic of mycetoma. My name is Carole Minlekib, and I'm a PhD student of Professor Doudou, who is the head of Parasitology and Mycology Department at Gaston Berger University of Saint-Louis. So I will first give you a brief definition of mycetoma. Then I will talk to you about how we are proceeding with the diagnosis. And finally, I will present you some of the cases we've selected. Mycetoma are a slowly progressive infection of subcutaneous tissue, which is characterized by the discharge of grain and purulent material through sinus. The disease is endemic in tropical and subtropical countries such as Senegal and Sudan, and it caused either by fungi or bacteria. Due to there are many etiological agents, biological diagnosis is important. Moreover, actinomycetoma and eumycetoma must be distinguished. So here we are working in a group team, including dermatologists, histopathologists, mycologists.
Carole, we lost your voice.
Can you hear me?
Yes,
Hello?
yes, we
Okay.
can hear you now.
So I was saying that for each patient with clinically suspected mycetoma, samples are collected ultrasound guided, either by thin needle aspiration, skin or surgical biopsy, in order to have viable grains, which is very important for the diagnosis. The surgical biopsy is done by the surgeon, the thin needle aspiration and skin biopsy by the dermatologist, and the ultrasound is done by the radiologist. As you can see here on the movie, we were collecting black green for a patient from a patient who has mycetoma on knees, and the lesion was discharging black green. Usually the sampling is done into two tubes, the first one containing 10% formalin and the second one containing serum saline. The first tube containing formalin is transferred to the histopathologist, who will perform histopathological tests, particularly hematoxylin eosin staining, and depending on the granulomatous reaction of the tissue and the microscopic appearance of the grain, he will give an indication or sometimes the responsible species. The second tube is transferred to the mycological laboratory, where we are performing mycological and molecular tests. The first step here is to wash the grain, and depending on the color, the grain are washed with or without gentamicin. White and red grain are washed carefully with serum saline only, and black grain are washed with serum saline containing gentamicin. The direct examination is performed with a drop of potash to note the presence of hyphae and vesicle. Sometimes it is easy to see the hyphae, as on the image, especially for the fungi, which is larger. We had also some cases where we didn't see the hyphae, as you can see on the image in the middle. The culture is done on sabro-destrose agar, sabrochloramphenicol, or Lowenstein-Jensen media, depending on the color also of the grain. They are incubated at 25 and 37 degrees. And as you know, there is some species that, such as Majorella grisea, which is not growing at 37 degrees. Then we are proceeding with the microscopic examination of the colony. But at this step, it is usually difficult to identify certain species, and that makes the molecular diagnosis very important. So for the molecular test, the DNA either from grain or culture are extracted using Zymokit, and then we are proceeding with classical PCR, targeting ITS region, 60S, or heat shock protein 65 chain. We are also performing real-time PCR to identify strain belonging to Actinomyra and Streptomyces genus. We have also sequenced strain from some patients here in Dakar, where we were able to identify 2 species of Majorella mycetomatis and 1 species of Mycosporum odwini. Here we have 1 patient of 26 years who had polyfistulized tumefaction of the finger with the discharge of red grain. Histopathology identified Actinomyra pelletieri as the pathogen, and on culture we observed pink colony that was heat shock protein positive and Actinomyra genus positive. And this patient was treated with Bactrim, and he is healed now. As you can see on the image, the finger is, we don't have any fistula. The second patient has had, of 24 years, had the tumefaction on his knees with discharging of black grain. Culture and sequencing identify Majorella pseudomycetomatis as pathogen. And in summary, over the last few years, we have made great progress in diagnosis of mycetoma by working together as a team. Thank you.
Yeah, thank you very much, Carole, for this nice presentation and about your experience with mycetoma in Senegal. I think we met some time ago in Senegal, where I visited your center with Professor Doudi and others. It was a very nice setup there for mycetoma diagnosis, as well as the treatment of mycetoma. And I hope we're going to continue doing this good work for the diagnosis, the management, and the care of mycetoma patients. I would like to say that usually, I'm very glad now we are doing the ultrasound-guided aspiration cytology for the diagnosis of mycetoma. And again, this is a very, very accurate method where you identify the grains, and then you target them by your fine needle aspiration technique. I would like to say that the use of a skin punch biopsy, it is not very useful. It cannot yield a lot of material for diagnosis because, as you know, mycetoma is a deep-seated mycology, and it is deep-seated within a very fibrous tissue. So the punch biopsy is going to take only part of the skin and will not reach the fibrous tissue in the deep subcutaneous tissue and the deep structure at that site. So please do not use this punch biopsy because it's not very useful in the diagnosis of mycetoma. Thank you very much. Back to you, Samira.
Thank you, Prof.
Dr. Abdullah is there.
Yeah, okay. Dr. Abdullah?
Thank you very much, Professor Fahar and Dr. Samira, and I would like also to thank Mr. Carole for this nice presentation. I'm sorry, I will make it very short because I am in a public place. But what I would like to insist that the grains is very useful for the differentiation between bacterial and fungal mycetoma, of course, but the culture and the grains need to be also tested by PCR and sequencing as mentioned in the presentation just to confirm the species. Thank you very much.
I would like just to comment about the importance of the culture and the histology for the diagnosis of mycetoma. In many centers, these molecular diagnoses are not available. So the clinicians and the pathologists are left only with this culture as well as histopathological techniques. What do you think, Professor Limia, about this?
Sorry, I missed what you said, Professor. Can you repeat it? Sorry.
Yeah, I said it is very good to have the diagnosis of mycetoma using molecular techniques. PCR
Yes.
now it is routine for the diagnosis of mycetoma. But in many endemic areas where mycetoma is common, this technique is not there. So a lot of people rely on the histology as well as the culture to establish the diagnosis. What do you think about this?
Yes, that is very true because as it's like you said in remote areas, so there isn't the luxury of the molecular techniques. So I think the gold standard of histopathology and culture is still proven to be of value, of course. And your comment about the punch biopsy, yes, I do agree because as you said, they are deep-seated, and this fibrous tissue around the granulomatous reaction will not be yielding a lot of information. Plus, I'm always worried about the recurrence, so the margins are important. So if you have a large biopsy, I think this will prove to be of good diagnostic value for the patient. So still histopathology and microbiology will still remain the gold standard. Thank you very much.
Thank you.
But thank you, Professor Limia. But I think we need to make it very clear for everyone that histopathology, if you have a good suction, if you have a good stance, you will definitely see the fungal hyphae within the grains. And this is very enough to prove that this is a fungal mycetoma. And if you have experience in area, maybe you are going to have also a good experience in telling that most likely this will be, for example, Madurella mystomatis or Actanomadura maduri or Actanomadura belletrii, just by looking into the grain structure under the microscope. So this is very good for differentiation between bacterial and fungal mycetoma. And also the culture is a very good gold standard for, because once you have a mycelium or you have a bacterial biomass, once you put it under the microscope, you can easily distinguish between the bacteria and the fungi. Fungi usually have a broad septate hyphae, and the bacteria, it's always fine filaments, and you can also stain it by gram stain, and mostly it's a gram-positive bacteria. But for the differentiation of the species and differentiation of the bacterial species and the fungal species, here the molecular biology comes.
Yes.
But for remote area, what the clinician need to start the medical treatment, the correct medical treatment is just to know at least, is it fungal mycetoma or a bacterial mycetoma? And this can be done in a very single and very remote laboratories just by training the people in doing a very good culture examination or histopathological examination. But for the differentiation of the species, definitely we need to go beyond that, because even for the today presentation, we saw some slides, and you can just see sterile mycelia without any spores, so you cannot differentiate the fungal species by looking into the culture. Thank you very much.
Thank you.
Thank you. Dr. Augustine, I see that you have a raised hand. Unmute yourself and go ahead.
Yes. For me, it is not a question or a big comment. It is just to let know all the team, essentially the Senegalese team, that I'm very, very proud to see this presentation and to see how things are moving, because I see that the team in in Senegal, in Dakar. It is a multisectorial team, and a success to have an early diagnostic, for the first patient they present, and the treatment was with success. And this one are some situations that encourage people for to accept the treatment. And my need is, and my request is for all the team in Senegal to be available to help the French country, because we know that MRC is very, very good for all English country, but for French country, we don't have a center where people can be trained and people can be taken in charge. And for that, I'm happy and I'm proud. I encourage the team, and I think Professor Fall could help more again to improve this center. Over to you.
Thank you, Professor. Yes.
I would like to add a new modality for diagnosis of mycetoma, that is the artificial intelligence-based diagnosis of histopathological slides. And this is a PhD done by Dr. Hiam. She just finished her PhD. Now she's doing her postgraduate studies in France. She managed through this technique to have a model to diagnose histopathological slide using this. We are not going to get rid of the histopathologist, Professor Limia, but that will going to help people working in remote areas where they can just put the slide, and then the system and the program will tell us, is it actinomycetoma or is it eomycetoma? And it was very tedious work. She saw a lot of slides, sections, and she trained the module. And then after that, she tested the module, and that was a very good result, almost more than 95% accuracy for this technique. So I hope this technique will going to be available for other people in areas where there is no histopathologist. So the lab technologist can prepare the slides, put it under the microscope, take photos, and then the system will going to tell you it is actino or eomycetoma patients. Thank you very much. What do you think about this, Lima?
I was just going to ask you, so will it only tell, which brings us back to Professor Abdullah's comment, we need to know initially so that the clinician can start the treatment, is it bacterial or fungi? With this AI, will it give a thorough histopathological report? Well, it will only, will it talk about the reaction? Is it a type 1, type 2, type 3 reaction? According to Dr. Hiam's work, well, it will just say if it's bacterial or fungi.
Yeah. So at this stage, she will tell us only this is a bacteria or fungal diagnosis.
Okay.
And hopefully in the next step, we're going to look into the different types of tissue reaction, type 1, type 2, type 3 reaction. But that is not important for to start the treatment patients. So we need just to see what is the underlying pathology, what is the organism there, and then we see how can we do. But how I am glad to say that it is more than almost 96.4% accuracy, the test.
Excellent.
We're going to ask Hiam to give us a talk about this.
Actually, Hiam will be joining us in the upcoming weeks. We'll just let people know. But there are some questions in the chat. Tambo is asking, how can we make access to equitable diagnostic tool for timely detection and care? So, Prof, can you hear us?
Sorry?
And Carole, are you still there? There are some questions in the chat.
Yes.
So Tambo is asking, how can we make access to equitable diagnostic tool for timely detection and care?
And I cannot hear you here, Samira.
Just if
Okay.
you check in the chat contain questions, several questions. This is the first question.
Prof, can you hear me?
Abdullah, can you answer this?
Can you answer?
I
Yeah,
didn't hear.
I think
No, I cannot.
equitable like access to diagnostic tools. I think it's only through training, because if you have a good trained laboratory technicians and laboratory technologists and pathologists, you can easily diagnose mycetoma at early stage. As Professor Fahal said already, that there is no importance in the early stage to differentiate between the different tissue reactions, because this is of no value for the diagnosis. For the diagnosis, we need to know, is it mycetoma or some other pathological conditions which looks like mycetoma, like chromoblastomycosis and other subcutaneous mycosis. If it's mycetoma, it's very easy to see. If you have a good biopsy, you will see grains. And if there is a grains, you need just to know whether it's a bacterial or fungal grains. And this can be done at the histopathological level or in the microbiology lab. So how can we help other people? Only by training. And we are open, and we said several times, I will keep saying it, anytime if you need any help, we can conduct an online sessions, training your staff in how to diagnose, how to look into the slide, how to prepare a good quality slide for histopathology and also for microbiological assessment.
Oh, thank you so much, Dr. Abdullah. And I think, Lubna, you wanted to add something? I've seen that you have a raised hand.
I think
Hello, good afternoon. Also, as Dr. Abdullah said, we need a well-trained laboratory personnel, and also and if there is the develop of a point of care test for diagnosis of mycetoma, it also help. And also when we do a community awareness or education, and also it helps for diagnosis of early diagnosis of mycetoma. Also we need conducting of research, and also to know what are the epidemiology of mycetoma. Are this area is endemic with mycetoma or not? And also developing of telemedicine for contact the area, for example, an endemic area, rural area with a center area for new cases or for rare cases of mycetoma. Also, this will help for diagnosis of mycetoma. Thank you.
Yeah, this is very good. Thank you very much, Lubna, to raise the issue of telemedicine. Now, through this technique, you can have a lot of advice about the management of mycetoma patients. Now we can use the technique for having a history examination of the patient. Even you can talk to the patient, and then after that you can advise on the proper treatment for such patient like this. And now, as you know, that at also we used to have this telemedicine facility where the patient are seen in the village, and then they communicate the information to the center in Khartoum for follow-up and for diagnosis and treatment for these patients. Thank you, Lubna. Thank you, Lubna, for giving us this opportunity to talk to us. Lubna, she is going to move next few weeks to Japan, Nagasaki University, to pursuing her master and PhD there. Congratulations. I wish you all the success in Japan and in Nagasaki University. This is one of the top universities around the world, and it is one of the top universities in Japan. And I'm sure you're going to gain a lot of information, a lot of science, and a lot of activities there.
Thank you very much, Professor. Thank you.
Congratulations, Lubnan, and wish you all the best. Also, a question in the chat, someone is asking about how the mycetoma samples could be conserved before sending to lab.
If I may answer, sorry.
Yes.
If you mean histopathology, it has to be within 10% formaldehyde, okay, and submerged, because otherwise it would not be fixed and the tissue would be autolyzed. If they mean histopathology.
Sorry.
Yes.
Okay.
No, go on, please.
Okay, here for the mycological test, we are conserving grain in serum saline, and which is put in the bag with ice pack and transferred quickly to the laboratory. And for the histopathological, also they are put in 10% formalin.
The Mycetoma Center had produced a lot of SOPs. SOPs for the transfer, collection, and processing of this material for diagnosis of mycetoma, and even for the storage of this in the biobank as well. So please see the Mycetoma Research Center website under the publication. Under that, you will find a SOPs section, and all these are available in that section.
So thank you, Prof. Does anyone has a question or comment? Feel free to unmute yourselves and go ahead. So, Prof, I see that there are no more questions in the chat. We've answered all of the questions in the chat, and
okay.
Shall we move on to the next part, or?
Yes. You are the organizer. Please.
So can you see my screen?
No, Samira.
Oh, okay. So now can you see it?
Now we can see.
Now you can see it, okay. So just. So I will be taking you today through the Mycetoma Young Researchers Association, MYRA. So I am Samira Mohammed. I'm a pharmacist. I'm a research assistant at the Mycetoma Research Center, and I'm also a teacher assistant at the Department of Pharmacy Practice and Clinical Pharmacy. And I declare no conflict of interest in my presentation. So through my presentation, I'll be taking you through MYRA, its progress. So the Mycetoma Young Researchers Association, it was launched in 2002 with the aim of gathering young researchers interested in research in mycetoma. So MYRA envisions a world where mycetoma is no longer a health burden. Why? MYRA envisions a world where mycetoma is no longer a health burden by advancing research, raising awareness, and developing effective treatments. All of this to improve the quality of life for patients affected by mycetoma. Our mission is to foster a collaborative and supportive environment for young researchers dedicated to studying mycetoma. We empower them tools and resources, networks to conduct high-quality research. And all of this to contribute to the global fight against mycetoma. So our objectives are promoting research, networking, community engagement, and capacity building by offering workshops and trainings, internships to young researchers. Also advocacy and awareness. So the activities of MYRA are mentorship programs for young researchers and junior researchers, conferences and workshops, and research grants and scholarships. Collaborative projects. Before I go to the membership, to MYRA membership, I'd like to highlight the point that MYRA is a multidisciplinary association. So MYRA is open to young researchers and students with interest in mycetoma research, for healthcare professionals and scientists who wish to mentor and support young researchers, and also for institutions and organizations involved in mycetoma research and treatment. So regarding the so we launched MYRA back in November 2022, and presentations within the medical sector, entrepreneurship, and the launch day was attended by honorable guests from the WHO, and we had some guests from the Arab Research Scientific Federation. Also, we conducted a number of workshops. We had a workshop in research methodology and biostatistics in January 2023. We had a laboratory diagnosis workshop in March 2023. MYRA also attended the Karma Scrubs event in March 2023, and this event was aimed at increasing the awareness of medical students, of students within the medical field, actually. And we were there to introduce research as a career. And MYRA in action, we have been in action since March this year. We're working on several projects, and the Mycetoma Research Club is the hub for the initial dissemination of our results. So here are pictures from the launch day. And the picture on your left is from the event, from the Karma event. And as you can see on the right is Dr. Augustine. And this was also from the event. So you can follow us on Facebook or through the website mycetoma.edu.sd. You'll find a section for MYRA, and the registration is open for everyone interested in joining. We're happy to have you all among us in MYRA. And thank you.
Thank you, Dr. Samira, for this presentation, and this topic is now open for discussion. If you have any discussion comments, please go ahead. We are waiting to listen from you.
I see Dr. Nidal, you have a raised hand. You can go ahead.
Yes, good evening. I'm Dr. Nidal. I'm from Sudan. Recently, I'm located in Saudi Arabia. I'm founder of Qalam Cafe, but I have some questions regarding the presentation from strategic view. Is this something that I've already approved, or I can comment on your objective, mission, and values?
Okay, you can comment. Yes, go ahead, please.
What I have observed during the WhatsApp group and the presentation that you have multi-country members involving in the program. So I think it should be appear also in your mission and vision. Also, you are conducting multidisciplinary research. It should be also appear in your mission as a main value that you are providing to enhance quality of life. And for the objective, I think it should be broken down from both vision and vision. It's just what I observed from the presentation. I hope it's valuable. Thanks.
Thank you very much. Thank you very much, Dr. Nidal and Samir. I think the aim of this presentation is to showcase about the activity done by MYRA during the last few years, but we would like to have more ideas. How can MYRA be a useful platform for training of young colleagues? We are not looking only for mycetoma interested groups. We are looking for everybody interested in research and promotion himself or herself. So MYRA is open for everybody, but those who are interested in mycetoma will have some priorities there. But anybody can join this association, where you can have a lot of funding opportunities, research proposals, and a lot of activities. So I would like the floor to give us more idea on how can we improve the quality of the work is done or going to be done by MYRA in the future. Any suggestion? A part of the improvement of the vision and mission and objectives.
I think I can help on that.
Yes. Hello. Can you hear me?
Yes.
Yes.
Yes. I would like to suggest the addition of more, insight about publication of not publication, before publication, choosing a topic for science, as we are starters in research itself. Sometimes we encounter as clinician, we encounter some weird or odd cases, but we are unable to suggest or give like a proper research paper. So I'm asking about maybe it's a research methodology thing, but I would appreciate if you can host a session of scientific writing, choosing a topic, the difference between like case study and the sizes and so on. I think it's clear, right?
Yes, Abdullah.
Thank you very much. I think it's a very clear presentation, and it is a good initiative, and everyone actually read it. And as Professor Fahad said, and also as Dr. Sabih suggested, that it should be open for not only for mycetoma, but for anything supporting the mycetoma research or research in general. And it also, we would like to remind everyone that we also launched or suggested to launch a discussion group. This discussion group could be can be also hosted and supported by MYRA, and also it could be used separately to teach people, teach researchers, teach students and young researchers how to conduct research either in mycetoma or in other fields related to mycetoma. including the very basic things, which is, it could include the research methodology, how to select a research topic, how to formulate the research hypothesis, how to proceed with all steps until you get your research published. And also at the same time, if there is anyone struggling in any technique, whether it's histopathological or it's microbiological or molecular techniques, and he need any help, also we can support. And I think this is a good platform for young researchers to join together and find the gaps and find the problems which need help, and find the problems need to be solved, and they can suggest it to the other people, and they are very welcome, and we are very ready to support them in any type of research and support they need.
Thank you, Abdullah. Thank you very much. As you know, that MYRA is going to have a small group, very focused, and that will call the Mycetoma Research Tutorial, where a group can meet an expert and express the difficulty they are facing, and they find the solution from this expert. So from next week, we're going to have this Mycetoma tutorial group, and this will be a small focus group for a specific area, and people can have more in-depth study or discussion or help or assistance. For example, you can hold a session on how to choose a topic, how to write a case report, very simple case report. How can we write it and how can we publish it at this site? And as Abdullah mentioned, that there'll be a lot of things you can ask about certain techniques, bioinformatics or whatever. So expert people, they will come in and discuss with you and will help you about this. So just what I would like you to join the MYRA group. There is a website at the Mycetoma Research Center website, and you can, from there, you can fill the registration form, and then you will be part of the group, and you will be able to access a lot of resources and activities. Thank you very much.
Prof, if you allowed me to add.
Yes, please.
To Sabih, we are early career researchers. I am an early career researcher, and yes, we need mentorship, and so that's why we launched MYRA. And as Dr. Abdullah said, we are having a discussion, a mycetoma discussion tutorials or mycetoma tutorial groups. So, and it will be in action from next week. Regarding how to join, there's a registration form. I'll be sharing the links through the WhatsApp group with you. I see, Woosen, you have a raised hand.
Yeah, Samira. So I couldn't find the membership joining link. So maybe don't forget to post it after the Zoom meeting.
After the meeting, yes. I'll be sharing the link immediately after the meeting.
Thank you.
We always find people in this session and also in previous sessions of the Mycetoma Research Club asking about how can they get help in doing research or in solving certain problems, but still we are waiting for people to approach us. I think the WhatsApp group is not only for just announcing the next topics for each research club meeting, but also for sharing any information related or any request related to support the Mycetoma research. So don't shy, don't hesitate to contact us, even in the private message. If you have any issue, we are ready to support you online. We can also, maybe in the future, we can be able to visit your lab, make a small training, or we can also conduct an online dedicated training for your staff at any time in any topic related to mycetoma diagnosis. We have a very rich group which contains pharmacists, pathologists, surgeons, dermatologists, microbiologists. So it's a very diverse group. So don't shy, don't hesitate to contact us, even if you are a very senior staff. We learn from our like previous years working in different universities that sometimes people are shy of asking questions, but once they go into this training atmosphere, they feel that it's very easy and many things can be fixed. So you are all welcome to ask questions or suggest any training or ask for any assistance. We are very happy to support you and the staff in the Mycetoma Research Center, and also the people joining this research club are very welcome and very welcoming actually to support anyone who need any kind of support. Thank you very much.
Thank you so much, Dr. Abdullah. And this is actually why we launched the research club in MYRA, is to work collaboratively to increase our understanding. So, Prof. Tahir, would you like to add something? Anything?
No, we are very open. We are very much happy to see people are interested in mycetoma and other research in general. And we are very much supportive to do a lot of support, assistance, to give advice, to help in writing up the case reports. And we're looking forward for having some grants. I can see from the chat somebody asking for a grant for research. Yes, there is a lot of grant available, but we need to know how we can get these grants. It is there, but how can we fish it? How can we get it? This will be the problem. And for you as young researchers, you have a big chance to have more grant for your research and for career development. Being in Africa, being in one of the low-income countries, again, this is an advantage where you can have a lot of grants for scientists in African countries, in low-income grants. In low-income countries, they can get a lot of grants. So it is important thing. It is you know how to can get the grants, and one of our session will be devoted for how to write a proposal and how to can get the fund for your research.
Thank you so much.
The most important is to ask for help, to ask for support, and people will be very much willing to give you the support and the assistance.
Well, thank you so much, Prof. And I think there's a question in the chat, Prof, saying that Tambo is asking, please let's explore cross-cutting research related to mycetoma and comorbidities burden in low and middle income countries. So do you like to comment, Prof?
Yeah, you comment. You're gone.
Of course, Tambo will be working on this, exploring the cross-cutting research, and a lot is coming.
Could you just elaborate on what you mean by cross-cutting research?
Tambo, would you like to?
Yeah, I think they mean to get this a grant. It is to have a good collaboration. You start to collaborate with people working on mycetoma endemic areas, and then you can collaborate with overseas. From the north, people are having the labs and having the activities there, and then you can have this. You can write a grant together, and after writing the grant, you can apply for a lot of funding bodies. Very important, you have to think of, you have to improve the quality of the lab you have in your center. You are not going to depend on the people from overseas, from the northern part of the globe, to support you all the time. You cannot send your biological material for overseas for diagnosis and you can come back of this. I think we should start good infrastructure capacity building. We have to train our staff to do these labs activities, and we have to work together. So I think my advice is to think about how to improve the quality of the labs and the research centers we have in our areas.
Rambo, if I think it's better to unmute yourself.
Samira, please send us some grant site where people they can see the grant, and then we can see how can we apply for these grants.
Okay. Okay, good. And just to highlight that multiple grant opportunities are being shared on the WhatsApp group. I see that some members are also sharing some opportunities. So does anyone has a question, a comment?
Limia, do you have any comment?
No, thank you very much. The idea of MYRA is really inspiring, and I'm sure it will help a lot of young researchers. And I think the ground rules are there and the foundation is there, and just people need to collaborate with them to help them, and we are ready to help in any way. So that's really excellent. Thank you very much. Shukran.
Thank you.
Thank you.
Okay.
So, Prof, I see there are no more questions. And
Yeah, okay.
Dr. Augustine, would you like to say something?
Yes. As usually, my interest is very, very big, and I'm happy to see that there are many young researchers, and there are things even in Senegal. Even when I'm reading the message from Tambo, I see that the interest is big, and then I encourage young researchers, as Professor Fall says, and I need really researchers to be in touch with the national program and the Ministry of Health, because all those things we are doing must be in interest of the patient, and the Ministry of Health is in charge of patient, and together with the Ministry of Health and WHO, we can be strong and we can make more difference. And for that, we are supporting MRC, and we would like to continue to support the team in Senegal to have a French pool, the French site to compensate the lack of French country to be supported. That is my comment for now. Over to you.
Thank you, Augustine. Augustine, as you know, he's in charge of the Mycetoma program at the WHO Afro region. So he's in charge of all the activities of Mycetoma in Africa. So I think we should ask him to organize for us a workshop on the diagnosis of Mycetoma, the management of Mycetoma in one of these African countries, where people can meet there and we can have a lot of experience exchange during that meeting. Or we can organize even multiple webinars for the diagnosis of Mycetoma in the region as well, Augustine.
Yeah, that's a good idea, and then we shall fight for fund, and then when we found fund, we can organize something. And all right, already with the NTD lab network, we start already to do something, and then I think next year we will have a meeting in Geneva for skin NTDs. All those opportunities, we will take it to go forward with Mycetoma issues. Thank you.
Okay. There is some suggesting that to have a translation to French of our website. This is a very good suggestion. I'm going to work on that.
Yes, it is a good idea, but the most good will be to have translation, because we can't be divided. We must be together. I'm fighting for French country, but I know that we can't want to be aside. If we are together with French translation and the English translation, it will be good. That's my idea. But we can start even when we are just meeting with the country speaker, the French country speakers. We can speak in French, but globally like this, I would like to have a translation and to give opportunity to both French and English country to exchange. That is my idea. Over to you.
Okay. Thank you very much, Agustin. Any comments from the floor? Ideas to improve the quality of this meeting and MYRA and other meetings? Hello. Yes. Oh, Karima, how are you?
Hello, Prof. I'm fine. And you?
Yeah, I'm fine. I will try to call you, but you're not answering your phone.
Okay. Thank you. Thank you all. I missed some comment because I have another saddle again. But I think it is a good idea. The idea of Dr. Augustine to have a French school or group to like the Mycetoma Research Center in Sudan. It is important to have such like in French speaking countries, because the language is often a barrier for French speaking in Africa researchers. So in my opinion, it is important to go to organize or to bring a network for French speaking countries researchers around Mycetoma. Also, it is also important to keep this group for all of us, because it's important also to participate to the English group, because many of the opportunities are in English. So it is also important to participate to this group. And secondary to have a small group of French-speaking researchers around Mycetoma. It is a nice idea. So I'm ready to participate to this as this group is organized. Thank you for all your comments. I missed the beginning because of a student agenda. So, but it is important to work together, French-speaking countries, as well as other countries, to fight against Mycetoma and all the NTDs, as well as all the fungal infection in general. Thank you, Prof. Fahal, for all your support. Thank you, Dr. Augustine, for your suggestion, and Dr. Abdallah, all of you. Thank you.
Thank you very much, Kadim, for this comment. I think we have to think how can we involve our French-speaking colleagues in such a discussion and a meeting. Thank you. Now to stick, Samira.
Yes.
Shall we?
Yeah, I think there are no more questions. And I see that in the chat, there are no more questions also. So Dr. Kadim, regarding saying that you missed the first part of the meeting, it's totally fine. You can go back to the recordings. Just you'll find it on on the Mycetoma Research Club in our website, in the Mycetoma Research Center's website. You'll find the recordings of all of the previous sessions, as well as you'll find the presentations. So I'd like to thank everyone for coming today, and I'd like to thank Dr. Carole for presenting their work and sharing their experience. Thank you so much. And I'd like to thank our participants who joined us and who actively engaged in these discussions. And I hope that you have a wonderful day, and we will meet back again in upcoming sessions. Thank you.
Okay, bye.
Thank you. Next time.
Thank you.
Thank you. Bye.
Bye. Have a nice day.
Okay.
Bye. Thank you.
Bye.
Bye. Thank you.
Bye.
Bye-bye.